Welcome to Science Frontiers, a space for discussions in the frontier areas of science and technology. I am your host, Binay Panda. Just imagine if the electric socket in each and every house is different, and you need to carry a gazillion adapters to hook your device every time you enter a house. Would it really be nice? And that's exactly the reason why a standard circuit was designed and developed that fits all kinds of plugs, at least in the same country. Standards help in achieving consistent, compatible, safe, and effective results. But unlike engineering, biology often lacks standards. Standards are especially important in synthetic biology, and this is for two reasons. First, the parts to conduct experiment originate and get assembled everywhere, all around the world. And second, the output from one experiment may become the input for the second one. And therefore, standards are very important in synthetic biology. In this episode of Science Frontiers, the third in the series on synthetic biology, we shall discuss about standards with our guest Pawan Dhar. Pawan is a professor in the School of Biotechnology, Jawaharlal Nehru University, New Delhi. Pawan, welcome to the show and thank you for joining us.
Thank you, Binay.
Thank you. First of all, can you describe to our viewers what are standards, particularly in the context of synthetic biology, and then describe what will happen if there are no standards?
Wonderful. Thank you very much, Binay. This is a very important topic with reference to biological engineering efforts. So standards are definitely the foundation of the engineering processes. And they are published documents that establish specifications and procedures designed to maximize the reliability of materials. The materials that you use, one needs to be absolutely certain that these materials are reliable. So reliability is one thing. And then second is of the materials. When you make the products, they need to be reliable. When you have the methods, they need to be reliable. So all of them, these three, the material, the product, and methods, they come under the standards. And they have to perform as has been described in the book. Consistency is very important in building the standards. They're the fundamental building blocks of engineering processes. And as you mentioned in the opening statement, it's only through the use of standards that interoperability is assured.
And what will happen if there are no standards? Although this this may sound as a moot question, but what will happen if we don't use any standards?
Oh, so it'll be a big mess because if there are no standards, let me give you an example. For example, you have tube lights in every house. So now if one tube light goes bad and you go to the market to purchase another one in replacement of the one which has gone bad, and then you realize that you don't have the tube light of that size. So which means that you have to change the entire electrical circuitry, the socket, to fit in another tube light of another size. And as you mentioned rightly in the opening statement, if you do not have the standards, then it will be very inconvenient. The entire engineering process will be very inconvenient. And every country or every state or every city will have preferences of certain sizes, certain materials, and it'll be a big mess because you cannot perform the engineering. You may be able to build a system, but when you need to repair the system, then you need to replace the entire system. You cannot repair it.
Absolutely.
So you need standards.
Absolutely. In fact, if I may just say here, that standards are so important in life, no matter what one chooses to do. In fact, some of you who have visited the countries outside of India, you would notice that the roads actually looks very similar, whether you travel 1,000 kilometers or 100 kilometers. The way the footpath is designed looks very similar no matter where you go. And in fact, these are the results of using standards. If you go to houses, the shape, the size of doors and windows are standardized. In fact, standards are absolutely essential to produce not just something which aesthetically look good, but also can be very utilitarian. Now, coming back to the example that I cited, Pavan, and you rightly said that the finished product is important, but what is even more important are the components and the methods. So it's not just the socket which has to be same all around the country, but the materials that are used in the sockets and the way those materials are used to produce the sockets. Now, in that context, what are the different components of building standards, especially for synthetic biology?
Great. Vinay, first, let me spend a few minutes in talking about the general processes of the components of building standards, and then we'll come back to the application of that into synthetic biology for audience to relate to what I'm saying. So first of all, due process has to be followed. A certain methodology has to be made by the experts who are part of the standard building processes. And second, there has to be certain openness, which means everybody who is interested in this process should be invited on board, and they should participate in discussing what kind of standards are needed and what is the process. It should be brainstormed and verified by people who are the stakeholders. Third is when you have a group discussion and when a lot of different opinions fly here and there, then a consensus needs to be built. So the third step would be consensus. And consensus is built on the basis of data and on the basis of some logical explanation of why do we need these standards and what is the application of standard. Fourth would be the balance, which means that people from opposing views also need to be accommodated. It should not be like a top-down approach that one person or one organization decides the building of standards and everyone is forced to follow. Here, the process of balloting, it should not be an overwhelming influence of one thought on a standard, but it should be a balancing act. And last of all, to my knowledge, which is probably the most important one from engineering aspect, is that once the standards are published, now suppose after a few months, you realize that you made a mistake, unintentional error, and the standards were not up to the specification that you imagined, the real use case in the community, then there should be a right to appeal. There should be a right to revision of the standards, and they're not cast in stones, but they're also evolving practices, the best practices. So that's as far as engineering process is concerned. When it comes to synthetic biology, that's where the challenges are, because the engineering steps, some of them are applicable, yes, maybe most of them. But when you talk of the application of these principles in the actual reality in biological systems, you have a totally different system. What do I mean by totally different system is, you have a system which is analog, which is non-linear, which has emergence as one of the property. The standards in the engineering systems were not built on emergence. They were built on a certain predictability. There was certain interoperability. But when you talk about biological systems, you see one of the biggest casualties is interoperability. That means the standard that you built in, for example, E. coli, we cannot transfer this to yeast. We cannot transfer this to human cells because it is very strain specific. It is specific to the culture condition, to the strain, to the metabolic state of the cell. And also, when you build these standards, these don't stay as consistent properties, which means again that, let us say, if you're trying to build a standard process of maybe purifying an enzyme and monitoring the kinetic parameters of an enzyme, now there may be 10 people working on enzymes, and they have 10 different methodologies. The trouble is that if all the 10 labs have their own way of isolating and purifying enzyme, then whose methodology will be taken as a standard? Because there needs to be a consistency. So that's the first casualty here. Consistency of the process is a casualty. So that's one of the bigger challenges. Second is when you are trying to build a truth table thinking that the truth table, which is a logical input-output condition, you land in a lot of problems there because the values in each box, now it's a quantitative truth table, it's not a logical truth table. The quantities in each box depend upon the activator and repressor concentrations, and they don't stay consistent, which means the value that you obtained, you cannot transfer. So theoretically, yes, it is possible, but practically there are many challenges.
Thank you, Pawan. And I think, I will still ask you for the sake of students, because we have a lot of students, our viewer, who may not actually have done their first experiment in SynBio. Can you give a very simple example of synthetic biology and tell us what are the standards which are either used in that experiment or can be or should be used in that experiment, universal standards across the globe, which will help students to talk in the same language? I look at standards as a universal language. It's like if we are speaking in English and somebody doesn't actually understand us because he or she can't understand the language that we speak in. Would it not be nice if the global audience, everybody else, have the same language? Likewise, the standards are the universal language. So can you give a simple experiment for the sake of our students who haven't done their first experiment in SynBio, that what exactly are you talking about?
Wonderful. Thank you very much, Binay. I think it's a very, very interesting question, and the implications of this for people who are recently initiated into this discipline. Well, when you talk of global language in standards, in biological standards, interestingly, we do not have biological standards which are globally applicable at the moment. We are the same phase where engineers were at the early 20th century, when they were just trying to build the standards and then trying to compose the system based on the standards. So we're in a very, very early phase. So what could be an example of, biological example from the standard perspective? So for example, if you have to create a part, let's say an E. coli part, a gene, and then you say that if I introduce this gene in 5 different hosts where it will be accepted, we know there's a compatibility, so the compatibility issue is not there. So the expression of the gene that has been measured in vitro, that is in all the input condition and all the output condition. Input condition, what I mean by that is the inducer concentration. At different concentration, you put the inducer, and then you have an output, which is in terms of the protein concentration. So when you have this, what you call as a data sheet, you also include the time lag, which means how much of time does this gene take after you add inducer to the system? How much of time does it take to? So is it kind of a time delay? So you make a data sheet, you make all kind of measurements and say that this is the standard biological part I have created in E. coli. Now, let us say this biological part, a certain gene, you're expressing in 5 different organisms. In E. coli, or in, let us say, go one step back, in the cell-free system, it shows a certain kind of protein expression based on the inducer concentration. The moment you put it in E. coli, the expression profile changes. The moment you put in organism B, organism C, or D, or E, its expression changes. So this is the trouble. In the cell-free system, it shows a certain expression profile. But the moment you go into another system, I was giving an example of an E. coli, the whole expression pattern changes. So again, the philosophical question here is, what is the need of building the standard if you cannot use the standard? So initially, people used a BioBrick standard. And then there was Berkeley group, there was Boston group. They were trying to build these standards, thinking that when they combine the parts, individually there will be a data sheet of each part, but when they combine the parts, it will have a predictable behavior. It did not happen. It did not happen. Now, the reason for that, of course, one is, of course, the analog system and the non-linearity. That's, of course, there. And also, in every system, the part has different pressures from the environment, from the ecosystem within the cell. That's of course one thing. But the second, another aspect here is that this data, which was collected early on, 2006 onwards, it is still being collected in the BioBreak repository. Well, people invented long DNA synthesis technology. So, which means early on, you had one part, all the data sheet of one part, another part, all the data sheet of that part, the entire input-output values. And then you had third part, fourth part, fifth part, sixth part, and so on. Every part had a data sheet. And they thought by combining these values, they can create an in silico model and predict what kind of circuit will be produced. It's logical, because you have done a good series of experiments. But when they combined, the overall output shape is totally different than what they predicted. That's one. Second, the long DNA synthesis technology caught up. So, which means what? Then this is very interesting here, Vinay. What does that mean? Instead of combining the data sheet of each part, now you have an ability to create the entire circuit and install it as a plugin. So you plug in a circuit on, you have a mega cloning vector. So you put maybe 20, 30 genes on this mega cloning vector. And then instead of bothering with each component singly, you put it as a block, which means that you should have a standard of the entire circuit rather than individual, because your individual values are not adding up, right? So a lot of work is going on in that direction.
It's very interesting.
But we don't have standards. Yeah.
So it's very interesting that standards of individual parts, then standards of assembled parts, which you are calling as cassettes. Now, let's switch gear. I'm afraid we have time for two more questions. So let's move about what are we doing, if at all there is any initiative in India about synthetic biology standards. Are we anywhere close to making our own? Has there been any efforts? And if not, why not?
Well, it's a very relevant question. Once again, Vinay, there needs to be foundational synthetic biology before people jump onto the applications, because they are not defining synthetic biology in a crisp way. So you can do metabolic engineering and market it as synthetic biology. That's what is happening, unfortunately. Synthetic biology is very clear in its mandate that there has to be standard biocomponents. You may not call it parts. You may say the components involve the genes, the modules, and the networks, which means the parts, the devices, and the circuits. At every level, you need standards, not just at the parts level. So in India, we don't have any such program to my best knowledge. And there is a big requirement to have, a funded program, which is going to fund the whole program for at least 10 years, because it's a long process. It's not a short process.
So the good news is that there is a scope to build a brand new program on developing, using, and standardizing the standards across the country, which is in tune. And of course, we are a part of the global scientific community, so which has to be in tune with what people around the globe are also developing. Now, the last question, Pawan, is, of course, is reserved for our students. So if I am a student interested in developing, using, or even learning about standards, where would I go?
Well, it's a tough one, Vinay, because right now, of course, you go to the funding agencies for, applying for a particular project for building the standards. But more than the data sheet, I think what we need is really a process standard. We need to first agree on the process. We just went through this, the enzyme example. Likewise, if you want to culture the cells, you want to isolate the DNA, you want to do southern blotting, you want to do western blotting. First of all, everybody should agree for a certain consistent process. And the purity of the reagents that you use, the kind of technology we use. Once we standardize our process, then the next thing, which is product, will have more reliability. So it's very important to do this, and someone has to make a start. So I would think of this more in terms of biofoundry. Yeah, so I guess
That's right.
that's the way to.
So that's an excellent note to end, and to give you the clue that what's coming up in our next episodes, which is Biofoundry, and how biofoundries can be used not just to enhance our capability in doing large scale experiments, but also how we can develop standards and engage the stakeholders like students and the researchers working in the area. With that, we would like to thank you all. Thank you very much, Pawan, and look forward to seeing you later. Goodbye.
Thank you very much, man. Very nice talk.